Buonasera, buonasera, benvenuti a GiovedìScienza.
Good evening. Welcome to GiovedìScienza, Science on a Thursday. This evening we will be talking about brains and the Down syndrome, and an Italian discovery for a possible drug-related solution for some of the symptoms of this disease. Piero Bianucci. Well, first of all, I would like to recall something which every now and again I happen to hear and which I saw of Diane's, a French neuroscientist's work. A very young child in the first few months of their life, up to 1 or 2, remember that they have about twice your neurons, so that they're very plastic and they're probably much more intelligent than you. This is the first reaction that we have. In other words, you are worse off the older you get. Yes, the answer is the alternative is much worse. So there's also some news for today, and recently, and this is an idea of Agliotti, so we try to give a bit of news before we move on to the conference. Today's news is interesting, and it is linked to Turin in a certain way, because it was illustrated in Cell, which you know is a very famous journal, a genetics research that was carried out on 35,000 people who either were autistic or were on the spectrum, or who are parents of children who are autistic. This research project, which is international and carried out by a multi-center consortium, also saw the participation of Italy, the University of Turin, and the University of Siena, as well as the Turin hospitals. I think the research is led by the Mount Sinai School of Medicine.
New York, credo. Ora, questo che cosa è venuto fuori in sostanza? Sappiamo che lo spettro.
Well, what was the result? Well, we know that the autistic spectrum is extremely varied in their various levels. There's very severe autism, which makes communication virtually impossible between the subject and the world, or the person and the world. Then there are other situations which are not as bad, where this is partial or impeded communication. About 1% of the population seems to fall into this group. And about 1% of the population would appear to fall into this category. I will talk about it later. But 1% seems to fall somewhere along the autistic spectrum, but luckily, not all in the severe form. Research started in 2015. They read the DNA using a new technique, which is targeted to coding proteins, in this case, the ones that are involved in neurogenesis and the organization of the brain in early childhood, and 30 new genes have been involved that are to do with the brain of the child. So all in all, I think we've reached about 100 genes that have to do with it. But it's not good news, because the more the genes, the more complicated it is to apply solutions. These genes do a lot of things. They interact amongst each other. They switch on and off like Christmas lights, and there might be 1,000 involved. So we're right at the beginning, but in terms of the understanding of the disease and its origin, or the syndrome, it is very important. Maybe we shouldn't call it a disease, but a syndrome. This is very interesting because, for instance, if we were to understand that it's to do with synapses, there are many hypotheses. Clearly, if we were to understand that, you can target the treatment for that. If, on the other hand, it were more of a matter of organization of the connectoma, well, then we could work on that. So there are various hypotheses. You know that mirror neurons too have also been suggested. Rizzolati's carried out a number of research projects, but apparently they're not the mirror neurons. So what we're doing is trying to understand this mechanism. Turing has contributed. We now are aware of 30 new genes that are involved in the autistic spectrum, about 100.
In tema di geni e di meccanismi di comunicazione del cervello che non
And now we have genes and mechanisms that don't work properly. But Laura Cancedda, we knew very little of the brain, don't we?
Sappiamo tantissimo, ma dobbiamo sapere molto di più, dipende da come la vuoi vedere.
Maybe we know a lot, but we have to know a lot more to answer the question. But we do know some things, but there's a lot more to be known.
vi invito a salutare con un applauso.
Laura Cancedda, whom I would like you to welcome with a round of applause,
È l'autrice di quella scoperta di cui si parlava prima, una ricerca
is the author of that discovery we were mentioning before, that is to say an Italian research project, and it concerns a drug which starts as a diuretic, which therefore has absolutely nothing to do with the Down syndrome, but was then discovered to not be able to cure, but at least to treat some of the symptoms. Laura Cancedda has graduated in drug technology, then had a PhD in Pisa at the Scuola Normale, and then postdoctorate at Berkeley, California. Now she's back in Italy, and she is at the Italian Institute of Technology, and she leads this lab where they develop this research with the help also of the Teleton Foundation. This research project didn't start purely by chance, but let's just say unexpectedly in 2010. I would say by chance, because I was invited to this conference, and at the time I was doing fundamental research on development of the brain development and mechanism. And before that, before my speech, there was a researcher talking about the Down syndrome. No, in fact, he spoke after me. First I spoke, and I said what I was doing, and then I learned some things on the Down syndrome, and then I got an idea, but only if, and because I went to that conference. In fact, my initial hypothesis was completely wrong, and then we refocused on it. As we will see in the presentation, this gives us an idea of how science sometimes or always proceeds. Unexpected results, finding what you weren't looking for. So the very little we know on the brain is not so little, but a very good starting point. Then at this point, I would like to leave Laura Cancedda the floor and the rostrum. Thank you. So that she can tell us about this. First of all, I would like to thank you, Professor Marchisio, and all the organization for having invited me. I'm always available to do these things because I like them. But when I was invited to this particular occasion, I immediately said I would, because I'd already heard about this initiative. I knew that it was something which was very high level. I hadn't really understood how good it was until a few days ago I started working on my presentation. And I'm a bit of a SWAT, so I went online and I looked and I said, let us see somebody's talk. And initially, I was attracted by this photo, so I started watching the video, and I said, I've seen this face before. I went back, I saw who he was, I saw that he was a Nobel Prize, and then I got a big question mark. I don't know if I can do things. Then I sort of said, I can do what I know how to do, what I normally do on these occasions. So I decided that I will be talking about how the brain works so that we can understand each other. And then I will give an overview on the technique that we use in the lab. And then I will also say something on the experimental results in my lab on Down syndrome. And then faced with such a well-educated audience, I decided to go a little bit further than I usually do, and to also talk about some of the future outcomes and also of what we have to do as scientists to go from an idea to its scientific implementation, and also, in the best possible cases, to transfer these results into clinical studies. What do we know about the brain?
Qualcosa si sa. Partiamo proprio dal...
We've said something, we don't know what, but let us start from the beginning. It's simply an organ like lungs or heart, but it's a very special organ because it, in fact, looks after the well-being, and I don't say survival, but I say well-being. The brain can weigh between 1.3 and 1.4 kilos in an adult, and the extraordinary thing is that it is formed by about 100 billion neurons, which is connected to tens of thousands of the others. So what is surprising is that it is a very complex organ. In fact, there are some areas of the brain that are specialized. For instance, the cortex, there are some areas like the occipital cortex, which is what I was studying in Pisa, which is the visual cortex, and which processes all the visual inputs, or alternatively the hippocampus, which is the part of the brain that more directly is involved in learning and memory. The brain is not only organized in different areas, these areas could communicate among each other to complete all the messages that come from the outside, decide what to do, and so on and so forth. This increases the complexity of the brain. So the question is, how does all this develop? And this is what we're studying. You must know that it's generated with a process which is called neurodevelopment, which is the process whereby from one cell you have the first networks of neurons, and in the end you have the brain as a whole. What we do know is that there are basically two phases of neurodevelopment, which come one after the other, but at a certain point they also coincide. There's a first phase which is basically developed by pre-established genetic programs, and then when the organs for sense develop, eyes or ears, what becomes important is experience, visual experience, auditory experience, and so on and so forth. So in the first phase, you have the first early connections, the initial connections, and then as time goes by and experience develops, we strengthen connections. This makes it possible for the brain to develop. Here there is a person, but in fact what we study in our laboratory isn't a human model, but that one. This is an example to show you. On the left is what a newborn sees. That is to say, when a newborn has not yet had a lot of visual stimuli. And so this is the genetics that has made it possible for the newborn to see what there is on the left. And then as an adult, it moves on, and experience is very important to remodel the brain. So it's a mix of genetics and experience. So the good news is that even if you have a non-standard chromosome series when you're newborn, it can change. But also, if you have a super chromosomes when you are born, it isn't necessarily going to work out well if you have negative experiences, because negative experiences can work differently on it. Let us see what the neurodevelopment is. All happens in this part of the brain, as you can see, where basically there are the initial cells, which then multiply and then differentiate. They become the different type of neurons. And not only neurons, there are also other cells in the brain. Once they have differentiated. We've said they're different parts of the brain. So they start to migrate. That is to say, they move in all the various areas of the brain and start to populate. Now, what we do know is that if something negative happens to each or any one of these phases, you may have the onset of diseases. So we've seen that there are all the connections amongst the various parts of the brain. The point is that how do they speak to each other in small networks, but also from one to the other area of the brain. They speak to each other because basically neurons are very special cells. They're polarized cells with a cellular body and dendrites. That is where the information arrives, and then there is this process which is called an axon here, very long, and that is in charge of communication. So a neuron wants to talk to neuron B. If neuron A wants to talk to B, the information will go through the axon, and the communication will happen in place which we call the synapses, where the neurotransmitter is released, the small molecules which help neurons communicate, and you generate electricity. So basically, neurons communicate through the exchange of this electricity. How is this generated? And you see here, because the neurotransmitters are linked to specific proteins that are channels, that when the neurotransmitter goes through, they open and allow the ions to go through, the ions that clearly imply a current. There are two neurotransmitters that are the most important, GABA and glutamate. Now, we know that GABA is the neurotransmitter that is in charge of communication, allows the flow, while there is the inhibitor neurotransmitter that will slow down communication amongst neurons. For example, if there's not enough GABA, there will be two communication, and you can have an epileptic fit. If there is too much GABA, then the communication will be silenced. And for instance, drugs that increase the GABAergic drugs will be considered drugs that you use if there is hyperanxiety. So you have to have a balance between excitation given by glutamate and inhibition given by GABA. We also know that in the case of the balance not being perfect, then you can have a number of diseases that are listed. How does it work? I've just said something about this, but let us move on to a second point. How can we study neurons? Well, first of all, we can visualize them, and here I've given you an example of neurons that have been cultured and can be visualized. Here you can see these as translucid cell bodies, and here we have to manage that we don't understand which one is the dendrite, which one is the axon. So a little time ago, well, quite a while ago actually now, researchers saw that there was a jellyfish that was able to produce a fluorescent protein. And through molecular biology, they managed to understand the gene that would code this protein and get, for example, neurons to express it so that they too would become fluorescent and they could be seen. And this is an image that we took in the lab we did of some in vivo neurons that produce this fluorescent protein, and as you can see in this case they are translucid, but you can see very clearly the dendritic tree, the axon, and so on. Well, this should be this green fluorescent. This is blue because, in fact, we started with the green fluorescent one, but then with techniques of molecular biology, we managed to have a range of fluorescent proteins that experts can use so as to differentiate one neuron from the other and visualize them.
Diciamo differenziare un tipo da un neurone o di un altro e visualizzarli meglio. Semplicemente guardando dei
By
neuroni che
looking at neurons that express this fluorescent protein, here you can see it in black because in the image I wanted to show them, they're colored, but just looking at them you can see that there are neurons of non-modified mice, the wild type, the control, the standard, or the Down syndrome ones. Now, you can see that the Down ones have neurites that are shorter and fewer. And if you look more carefully, you can see that each one of these neurites, these dendrites, have fewer of what we call spines. That is to say, where the synapses are. So by simply looking at the neurons of modified and non-modified mice, you realize that the Down syndrome mice have fewer neurites and fewer spines.
a livello del numero di neuriti e a livello delle spine sinaptiche. Questo è solo una piccola
This is only a short sidetrack to highlight the importance of the green fluorescent protein. These three people won the Nobel Prize in 2008 for this discovery, and I always like to diverge here and talk about their lives, because their lives are very different and couldn't be inspiration. The first gentleman here is someone who of Chinese descent, very noble family, descendant of also an emperor in China, with fathers and sons who are great professors in American universities, all men, who had asthma as a child, so he stayed home and studied a lot. But he came from a family of people who had studied a lot. This other gentleman here who won the prize with them is the son of migrants, Russian migrants. The father was a guitar player, and his mother worked in a small shop that sold domestic appliances, and was brilliant with great intelligence, but a non-linear study. He studied maths and then decided that he wasn't interested. He wanted to do biochemistry, and then he wasn't interested in that. He left all that. He did all sorts of jobs, studied law, Russian literature, and then in the end he took a PhD, but then he got bored, became a Harvard swimming champion or nearly, but then he didn't do that. Then took a gap year, worked in his mother's shop, taught. Then he didn't like that, so he went back to biochemistry and won the Nobel Prize. And this one, who is the eldest, had a father who was in the military in 1945, took him to a village near Nagasaki, and in August he was there when the US launched the bomb. So at that point he had to study in Japan, where the universities were virtually all closed, and he wanted to study. So he decided to study pharmacy, not because he liked it, but because it was the only one still working there. And from there, though he was very successful, went to the US, and in the end, he too contributed to the discovery of the GFP. Now, let us go back and see how we can study neurons. Neurons can also be listened to. You can record them, because basically when they talk through this ion current. And you can record them electrophysiologically, basically to measure how many words or exchanges there are when one neuron speaks to the other. These are very sophisticated techniques, but they make it possible for us to understand how communication takes place. To do this, you use very advanced tools that I have the privilege of having in my lab, because I work at the institute that has always supported me, and these are very advanced instruments because the words that neurons exchange are very, very quiet. The currents are very, very small, so you need very powerful amplifiers or boosters to understand what they're saying. This is the setup, the classical one, and the neurons are sliced, and a mouse or others. Here you can see an enlargement. There's a microscope, and these are amplifiers to listen to them. And here, this is what you see when you look in the microscope, where basically you can see the pipettes for the recording. So what we're trying to do, we're trying to understand what these neurons are exchanging. One of them has been transfected with the fluorescent protein. So just by listening, you can realize that neurons of a non-modified neurons and the ones with the Down syndrome are different. They speak in a very different manner. And if we consider that each one of these here is an event, is a sentence that they spoke, you can see that the Down syndrome ones speak more frequently. So you listen to neurons, you look at neurons, but you can also study the behavior of animals. In our case, we were very lucky because there is an animal model, a murine model for the Down syndrome, which is one of the best of development diseases, because animal models, when you study a brain, clearly are very useful, but the shift from human to mouse or animal is not straightforward. But in the case of the Down syndrome, because we know what the cause is, that is to say the triplication of all chromosome or part of chromosome 21, we managed to produce a mouse that is very similar, as you can see here. In people with the Down syndrome, it's chromosome 21, while in the murine model, it is in chromosome, or free chromosome, which has part of the genes that are triplicated in people who have Down syndrome. In fact, this mouse echoes some or many of the health problems that people with the Down syndrome have multisystemically, and you can see it here. They're smaller. There are alterations of the face and skeleton, immunological changes, and heart defects. In terms of the brain, which is what we were more interested in the lab, they have an increased susceptibility to epileptic crisis, anxiety, hyperkinetic, sleep problem, and also have a slight degeneration, Alzheimer-like degeneration, which is very common also in people, in humans with the Down syndrome. Once again, you can study behavior, and this is an example just for you to realize how it changes. Here you see a non-modified mouse on the left without the Down syndrome. If released, looks around and takes a while to decide what to do, while this is a hyperkinetic one, and as soon as it is released, it starts going back and forward. So it is true that with all the caveats that an animal model can have, replicates many of the health problems that humans with Down have. Luckily, this model is quite good because to date there is no drug to deal with the cognitive symptoms of the Down syndrome. And so what we do now are treatments, early educational activities that can be very important, can give fruits or bear fruit in the future of the person if they have treated very early. Development is more standard, but it is clear that this is also based on the IQ of people, and are more effective in people with a higher IQ.
E sono più efficaci in persone
So a drug therapy that will support person with a Down syndrome with a low IQ can also be a help in early educational approaches.
educazionali precoci. Quindi a questo punto, appunto, al famoso congresso...
At that conference, I didn't know much about the Down syndrome. I knew it was the triplication of chromosome 21, but I discovered that in mice with the Down syndrome, there was a larger number of GABA-like cells. So at the time, what we thought was that given that there were more GABA cells and it's an inhibitor, neurotransmitter, there was too much inhibition, and that the mice had too many problems because they were over-inhibited and they were sort of a bit drowsy, slower, and this was the reason why probably they couldn't work properly. But also these mice should also have epileptic, be hyperkinetic, and have anxiety. And I didn't know that the Down syndrome, I didn't know much about it, but I knew that GABAs were an inhibitor. And it seemed a bit paradoxical that given that they were over-inhibited, they were hyperkinetic and epileptic, because usually that corresponds to stimulation when anti-anxiety drugs increase GABA. So the hypothesis was that maybe that there wasn't too much inhibition, but that the neurotransmitter was working the other way around. That is to say, it didn't inhibit, but excited. And therefore, the problems were due, yes, to an unbalance between inhibition and excitation, but not too much inhibition, too much excitation. So to see if this was true in adult animals, we sliced the brains of mice with a Down syndrome and simply listened to the neurons when they exchanged. Yes, these are the recordings that there were before, and we had realized that in the mice with the Down syndrome, the neurons were talking more, and this wasn't consistent with an over-inhibition. But then we carried out a direct experiment. We administered GABA to see whether it was inhibitor or not. You can see that in the non-Down ones, the GABA decreases the exchange. But when it is administered to animals with a Down syndrome, you see how they start speaking, indicating that possibly we were right and that GABA is not inhibitor but increased excitation. Luckily, that coming from the GABA world, I knew, and by luck, that there was a drug which is a normal diuretic and that is able, in the cases when GABA is excitatory, it's to switch it back to being inhibiting. So we did an experiment, which is what you do in a lab. We tried on these slices to administer GABA with the drug, the Bumetanide, because we know the effects that this has, and this is what happened. So administering GABA plus this drug to the mice with the Down syndrome, the GABA was no longer had an exciting effect, but it led them to behave exactly of animals that were not Down animals. So we asked ourselves whether this was enough to help these animals recover their cognitive capacity. How can you measure the cognitive ability of a mouse? It's not difficult, in fact. What you do is you carry out tests that are called cognitive tests, behavioral cognitive tests. You take the animal from their cage, you put them in an area like this, and the animal will try will find two different situations. The animals are curious and they will explore this object, then at the end of the session, back into the cage, leave them to sleep, and the day after, you test the animal again in the same arena, but in this case, one of the two objects has been moved. Now, the animal that has learned will remember the configuration of the day before, and being a very curious animal, it will explore the object that has been moved, while an animal that doesn't remember, that hasn't learned, will behave like the first day and will explore the two as if they were both new. So this is the quantification of data. As you can see, the memory index, the non-modified animals, have different behavior, while the animals with have a Down syndrome didn't get the same results. The result is that adult animals are treated with the drug that switched the GABA, recovered learning too. And you can see it here. This animal with the Down syndrome and this one, they were treated with Bumetanide for 4 weeks, and there is a recovery of their cognitive ability. We were very pleased of this because potentially this can be used in a safe way with human beings, because it has been used for 50 years as a diuretic. The only thing we wanted to be sure of is that to have some evidence that in human being there could be something which was similar. So we studied all the mechanisms and how it worked in the mouse, and we discovered that it worked because in the mouse there was the expression of this transporter, which is specifically inhibited by this drug, and that this transporter was produced in greater quantities. The same experiment, seeing whether the expression of the target was the same in human brains, and a biobank in the US gave us a slice of these brains, and we saw that in the Down syndrome ones, there was the cell defect, the same fault. So we had the result, and that is to say that our experiments were confirmed both in mice and humans. These are our preclinical data. We contacted the Bambino Gesù Hospital in Rome. We found a wonderful team, Dr. Vicari's, many years, but now we're really about to begin. They will be doing it clearly at the Bambino Gesù, the Dr. Vicari's group, a clinical trial for the use of this drug, not as a diuretic, but as a drug which will help the people with Down syndrome to recover at least partly their cognitive ability or potential. Let us see something about repositioning of drugs. The discovery of a new drug is something which is a very risky investment, both in terms of time and money. That is to say, you start with something with 10,000 compounds, and 15 years later, and enormous investment. If everything's all right, one will go on the market, because there are various phases. There are initial phases where you work on the compound, then there is the preclinical one where you test them, and then there are also the ones phase 1, phase 2 and 3 on volunteers, healthy volunteers, the very small cohorts of patients, larger cohorts of patients, and then if everything works, you in the end reach the market with one drug. So if you give an estimate, you need about $2 billion, 10, 15 years of research on 1 out of 10,000 molecules will survive. The cost of drugs for the central nervous system is more complicated for the CNS for a number of problems, which I'm going to summarize here. There are 3 issues. One is that when you carry out studies, clinical studies on people, for drugs that work on the CNS, it's very difficult to quantify the effect. That is to say, if you take an anti-cancer drug that works, it's easy to measure it, because if the cancer shrinks, you can see it. If there are fewer cancer cells, the drug has worked, and you understand that. But as far as a drug that must help with cognitive functions, it's much more difficult. You can carry out tests, behavioral tests, but it's not something that you measure. And this clearly has made it very difficult to develop these drugs. Furthermore, there is the problem of the placebo, because since it is not possible to measure, and because it acts on the brain, it is also possible the fact that you think that you might be better might generate a partial recovery of the overall health of the brain. It's very difficult to quantify.
Generale di salute, e questo è difficile da quantificare. È un effetto che si ha sia sul
This
paziente.
is both true for the patient and for the researchers, and because we know something, as was said before, but we don't know. There's a lot we don't know. The other thing is that if a drug, an anti-cancer drug, if you have that, you can imagine that it has the same effect on a mouse and on a human being, because a cancer basically can be replicated. It's much more difficult when you think about this to transfer the results that you have on the cognitive effect of a mouse to humans. Mice are not small human beings with a tail, so all this makes the discovery of new drugs extremely difficult. So over the past few years, on the whole in the medical field, but more so in the CNS, people started to do a repurposing of drugs. You take a drug that you know and you repurpose it, because this means that you skip all the phases. You have voluntary, healthy ones and volunteers, and you go on to phase 2, and it's faster and cheaper.
Quindi ovviamente gli studi di
So
riposizionamento
these studies have increased, and we're all very pleased, because it could be the first alternative for people with cognitive problems and a Down syndrome. It could be the first drug alternative. There are other clinical trials. We're not the only ones. But this happiness and enthusiasm, which I would like to convey to you, must be also considered, because there is a dark side to it.
perché anche il riposizionamento di farmaci ha un lato scuro, perché per esempio gli effetti
For example, the side effects are present because, for example, for a diuretic, the side effect has a problem. So if it's a strong diuretic, to recover cognitive ability can be something which in everyday life creates problems with drug compliance. Because at some point, people might decide not to take the drug and discontinue the treatment because of the side effects. In this framework, the drug industry has decided that drug discovery are being reduced, especially as far as the CNS is considered, because it's too risky. But there is a close collaboration between academia and drug companies based on the new uses of drugs, because the drug company enters when things aren't as dangerous. What are we trying to do? We're very pleased of the new use of the drug, but we know that if it might be a first and effective alternative, in the long term we will have to find a drug that acts on the brain but doesn't act on the kidney, and therefore is no longer a diuretic. So in cooperation with Marco Di Vivo's laboratory at ET, he works in drug discovery. We've been working now for 4 years, nearly 5, on a new drug that will act on the brain but is no longer a diuretic.
Ma che non sia più diuretico. I risultati non ve li faccio vedere
I
oggi
can't show you the results because the results haven't been published, not because I'm afraid of it, but because it wouldn't be serious faced with an audience to show results that haven't been validated by the scientific community.
Quello che stiamo anche facendo è
What we're
vedere
also
se
doing is to see if the other health problems that are associated with the Down syndrome, like anxiety or sleep, which depend on GABA, could benefit of this type of treatment. And we're doing this with an expert in another lab.
se un trattamento, questi
We're
tutti i
also wondering, these are all the treatments that we've been talking about on an adult animals. We're also trying to see, since the Down syndrome is a condition of neurodevelopment, if early actions could have
delle cose positive a lungo termine
positive outcomes in the long term
più tardi nella vita. Allora, il punto era questo, che
and on later life. So we tried the drug on cognitive problems and it works in adult animals, but we also already have tried it on the epileptic crisis and hyperactivity, and it doesn't work there. So we thought that for those behaviors, it's very important how the brain organizes, and maybe a very early action might help with these symptoms. But it doesn't work if the animals are adult. The last thing that we're doing is to try and find other mechanisms that could contribute to the severeness of the human pathology, because what is emerging, that animal models work, but probably they mime only one aspect of the disease or illness, but there may be other also animal models that mime another aspect. And so in the framework of clinical studies, it is important to try various animals. And in particular, we were very curious of doing the new animal model that hasn't got problems on the neurons, but this, with the Down syndrome,
gli animali hanno dei
the animals have cognitive problems. Maybe they're not the neurons, and maybe they could be other types of cells in the brain that contribute. The first model worked for the neuronal one, and maybe this animal model could work for something of other types of cells. And maybe we will have to use both the drugs together.
i farmaci insieme. Questo di nuovo non è
Again, it hasn't been published, so I will only show you something very quickly. It's microglia. We simply looked at it as we did first. These are microglial cells of mice without Down syndrome, and the one below is the Down syndrome microglia. Just by looking at it, you realize that there is an enormous difference.
ci si accorge che c'è una grossissima differenza. Allora, cosa intendo? E qui arriviamo
So what happens? And this is the last point I would like to raise today. It wouldn't be serious to publish the results, I said, because it hasn't been validated. What do I mean by this? It means that when researchers have an idea, and between that and when it is published scientifically, there are several steps. And I wanted to share with you all the work that me and all the others have to undergo to reach the bottom level, that is to say the top, which is the publication. First of all, you have to find funding because research costs, so you have to find money. I'm very lucky because I'm working in ITI. We have internal funding. Otherwise, you have to start to find things. A Teleton is a lot of them. But you have to apply, you have to have the project approved by experts, and so on. And then if your work is a quality work, then you do it. Then there are you have to have particular models, animal models with some are patented, and you don't want to cause as much. You have to have permission because you don't want to cause unnecessary pain. And then there's the work. And For instance, when I said that I wasn't going to release these data, it's because on sleep and anxiety, that's because we're there. Then you send it to a journal. And the journal has peer reviews and the editors. So that is to say, the great expert, who's called the editor, decides whether this is interesting or not and sends it to 3 or 5 experts in the field who read it and say, well, I think experiments are well done or not.
Vanno bene su questa rivista, ok. E il lavoro della microglia.
And microglia is here, so you can, right, we're very close to the end, and I'm going to have to show it. No, because once we've had the peer review, what has happened is that there is more work, because usually they ask you for more tests. So in the end, when you're very close to the finishing line, you get the frustration and the idea that in the end, although you're very close, you need time. And I'm not exaggerating because it's 4 to 6 years. And from the initial idea to publication, there are easily 4 to 6 years.
Tranquillamente dai 4 ai 6 anni.
But in the end, you succeed. And when you do something which has a potential clinical impact, then you wonder, how will this be translated in a clinical framework? And the answer is the same sort of things that you do for scientific research. You need patents, ethical committees, funding, and then work. And in this framework, the project of the new drug has reached this level. The clinical trial, I said it here, but the funding is much greater because you need studies, medical studies, clinical studies, very few patients at the beginning and a larger number of patients later. And this is where drug companies enter and cooperation between academia and drug companies. So in conclusion, I hope that today I have shown you how a brain develops, a healthy brain with various processes, and how you carry out research. And also, if something doesn't work, we know that the brain doesn't develop well. Here we have drug treatment, and the use of older drugs for new uses can be a shortcut. In the end, probably we will have to find a drug that acts like the old one, but no longer has the side effects that did. And from this point of view, and I repeat it, the work of academia and drug company together is essential. So, what can I do for you? And I hope that I have answered all the questions. Now, the question is, what can you do for us? I would say, believe in fundamental research, because basic research will help us find the best students and motivate them. We need 4 to 6 years to publish research. Research can be super frustrating because you don't get results, but when something does happen, it's wonderful. But in frustration, you must know that you're there, that you believe in our research is very important and motivates. And then you must guarantee the support of the foundation. Here I would like to thank Teleton, who believed in me and in our research. In conclusion, I just wanted to show you a series of images that my youngsters produced in the lab, which I consider works of art. Liquids, hippocampus on fire, lightning at night, rain at sunset, neurotic hippocampus, a new Andy in scientific art from Andy Schwetz, who was a Polish student who was in the lab, waves, brain forest, climbing the brain, neuronal rainbow, coral reef, which is my favorite, because I think these are GABAergic cells. So, in the end of the story, before sending you away, what's the saying at the end of the story? Fundamental and basic research has the potential to influence people's life, but you must trust and have a lot of patience. But everyone can become a good scientist. And the neuroscience is wonderful, it's beautiful.
Grazie a Laura Cancedda.
Thank you, Laura Cancedda. Really worthy of the round of applause. I see a hand up, so if there is a roaming mic, I would like to thank you because you were very, very clear. And then I wanted to ask you whether this type of drug, is it also suited for patients who have cognitive degeneration, so for example for Parkinson or Alzheimer's or any neurodegenerative disease? So, well, the interesting thing is that after the publication of the study which I mentioned today, which comes from my lab, and some studies on autism which come from Dr. Benary's laboratory in France, we saw that these mechanisms are shared not only by all of autism or other neurodegenerative diseases, but also to conditions which could also address some neurodegenerative diseases. There's evidence, very little evidence though, that also in the Parkinson that there might be something like that. But they're just starting. I would like to ask you if you have understood why there is a transformation between excitation and inhibition of GABA. It's that protein that I showed, which is called NKCC1, when we've seen that there is a production, an increased production of that protein, and that protein is basically a transporter of chlorine, which is very important for GABA transmission. So when there's too much in the cell, GABA switches and changes the way in which it communicates. What we don't know, which instinctively one might think, since there's an overproduction of this protein, maybe it is one of the triplicated genes of the Down syndrome, which is you have an additional gene, and maybe that is why. And it's not like that, it's not there. So there must be one or more of the triplicated gene that does something somewhere that then, in the end, causes the overproduction of that protein. I discovered at 57 that I suffered from Asperger's syndrome, which is a form of autism. What can you tell me about this and your experience? Well, in practice, it was far again as Bumetanide and CC1. I would say that there's one clinical studies of one person who was treated with the drug who had an improvement in some aspects of the syndrome. But as always, one clinical case, well, could be interesting. It is interesting, especially in a context where apparently it is shared by many people and by many facets of the disease or of the brain, but one clinical study is not. If there are any questions, please raise your hand. In the meantime, I have two questions. When you were talking about the going from the idea to publication, how much does the idea change? Does the idea the same? And the other question is, if it doesn't change a lot, what is there before?
Ok. Allora, quanto si trasforma?
Well, how much does it change? Well, it depends on each case.
In media direi si trasforma moltissimo all'inizio.
I think it changes a lot at the beginning. So at the beginning, it's very difficult to understand clearly what is happening, where we're going, and so on. But as my head in the US was saying, he said it was difficult at the beginning, but when you get the right path, you get all the experiments right, because it's very difficult for you to understand. But when you do understand, you go on, which is roughly what happened in this case here. So the answer is probably a lot, but in the first phase, and then it's difficult to make major mistakes. But to get to the idea, I don't know. In my case, it had a lot to do with chance. I get bored very easily, so I go to conferences, I read a lot, and that's how ideas come. Inspiration. I must say the truth. Yes. Looking at people or listening to people who talk. But that's how I don't know about others. Other questions? Maybe this idea, an inspiration. Right. I wanted to ask, people have been talking about tailored drugs and biodrugs. So how many possibilities are there in this field? Because a person I know who had problems all their life, but they can't stabilize them anymore with the drugs that are available. So I wanted to understand how many possibilities there are with these new drugs. Well, clearly, gene therapy is probably the future, but these tailored treatments require, by definition, a knowledge of the pathology which is very detailed, and this answers your question. In the case of the brain, we've said so far and right at the beginning, it's very difficult. So I believe that yes, there is a future because they're targeted and tailored, but I think that in the case of the brain, there will also be room for classical drug treatment, because progress in knowledge of what concerns the brain, and we know a lot, but there's still a lot to learn. One question there.
Come mai comunque un farmaco che in Italia non viene utilizzato, non è passato dal Servizio Sanitario Nazionale, ci sarà qualche motivo?
Something about the drug. Why has the drug not been approved by the National Health Service? Why is that in Italy? And why has it no negative side effects on normal patients if it has an effect on the GABA patients? So why doesn't it have any effect when it's given for the other indications to healthy patients or to patients that are otherwise healthy? First of all, congratulations for the question. Because this is a very advanced. The first one, why hasn't it been approved by the National Health Service on the drug? Well, the drug, you're quite right, in Italy, it hasn't been approved by the National Health Service, not because it's dangerous, because it is a very powerful diuretic, and it is difficult to dose it and use it, because if you use it too much, it gives very strong diuretic effects. It is used, but only in hospital. People who have heart problems because they're regulated with diuresis. So it's not that it's dangerous. It's simply that it is very difficult to use. And because there's a better diuretic that is furosemide, which acts more or less in the same way, but that has a much easier dose to use. Not because it's dangerous, therefore, because it was difficult. The second question is Yes, perfect question, yes. Because it doesn't work on animals without the Down syndrome. We are very surprised at this. We were. But this is something that not is a result which is not limited to our lab, but to all the labs that have been studying this. And there is a twofold answer, probably both. One is that I said that in animals with the Down syndrome, there is a higher expression of the target for the drug. And this, by definition, means that it's obvious that if that protein, which is the target of the drug, is expressed in higher levels with the Down syndrome, it'll work much more with Down animals than with non-Down animals. But also, they have some CC1, but NCC1, but they do have that in levels much lower. The second is, let me see if it's to do with GABA. Let me see if I can explain it. Basically, GABA can be inhibitor or excite if you change the concentration of chlorine, but very little, very small concentration when it's inhibitory, is that it becomes slightly more inhibitory. This is what we say in humans. So if you have a slightly more inhibited to a more inhibited GABA, from a physiological point of view, it doesn't make a big difference. The big difference is when it goes from excitation and you give a little bit and then it becomes inhibitory. But the physiological difference between excitation and inhibition is much bigger. I don't know if I've answered your question. The third question, I can't remember it. How does it pass the blood-brain barrier, the BBB? Well, the drug passes very little. This not only us, but all the other labs too, and we asked ourselves repeatedly why this was possible. This is possible that what passes is enough for the GABA physiology. There's enough, very little, to change something which is physiologically important. So maybe that's enough. And secondly, that there are indirect mechanisms outside the brain, or partly outside the brain, that change the concentration of ions, which means that then the functioning change, or, as happens in research, both things or another.
No, volevo chiedere, ma questo
I'd like to ask whether this drug, is it a treatment or do you have to take it for life? Or 3 months and then stop? It's a drug that you have to take for life, in mice at least. It's a treatment. As soon as you discontinue the treatment, they revert to what they were. The good news is that it works in a super acute manner. It works. You give him the treatment, and it immediately works on day 1 when they take the behavior, and you give it again to them, two injections, and already they're recovering. So the negative thing is that it's for life, but the positive thing is that you immediately have results. This in mice. I don't know in people. You can hope, but... I have a question. In the case of Tourette's syndrome, there's not always an involvement in cognitive aspects, but anxiety is very important. For example, I would like to know if the Tourette's syndrome is included in your studies or not. Not by us, but since it is a neurodevelopment issue, I think that probably somebody's working on it, but not our lab.
Una domanda qui. È anche difficile essere aggiornati su quello che fanno tutti gli altri.
It's also difficult to know what all the other labs are doing, but my youngsters keep me informed. Good afternoon. You were explaining the passage of information through neurons. that are electric impulses, and you were showing us the type of instruments that you use in the lab to study these electric impulses. I don't understand, since it's information, how do you convert the electric impulse into information? We don't convert it, but basically you give a meaning, for example, to the frequency, to the amplitude of these currents, so we know that when neurons speaking with that frequency, it means something. When it uses another one, it means something else, so on. With a certain amplitude, it means it does something, and so on. This is the idea. But these are groups in my research lab. I'm looking for possible treatments for neurodevelopment, but there are labs and research groups that are doing studying the coding of neurons. That is, try and understand exactly, by analyzing the currents, so what are the features of the language, so to speak. I still wanted to ask something else. Is it more important for you? Is the synapse more important or the neuron more important in development and in how the brain works in the future? Is it more important to work on neurons or synapses? Well, presuming or starting from the fact that the synapse is parting of the neuron, but I've understood your question. Probably it depends on the type of disease. For instance, for all a series of diseases, including autism, we also talk about synaptopathology, because many of the genes which we were mentioning before have been involved in autism. They code the proteins of the synapse. So the answer probably depends on the type of disease. But I would say that probably, given that the final point is the synapse, I think that it plays a key role. If there are no other questions at this point, very last one here. I've decided that it's going to be very last and very quick. You talk about stem cells. I would like to know whether in your case, is there possibility of applying stem cells? In abstract, of course, yes, because research is working on stem cells. That's the beauty of research, opening oneself to new ideas, but in the specific case, we're not carrying out any experiments in this regard. Thank you very much. I would like to thank Laura Cancedda.